introduce, and the idea here is to provide you some background about protein research and how it is actually affecting the research that we are doing right now, especially in the food context. Then I will go into the details that we can find in protein sequence, in the amino acid sequence, and what we can infer in terms of structure for food proteins. Then I will present you some physical framework to model protein behavior, and it will be a good link with the two lectures of this morning with polymer science and colloidal science. And in the end, of course, like protein in ingredients, in food ingredients, are far from the models, and so I will see how we can or try to make the link between the two, from model to real food systems. So I will start from the discovery of proteins. So the first person who identified proteins is an Italian scientist, botanist, and he noticed that when he was extracting, in the end he discovered that it was proteins, but basically he mixed it with flour, with water. I don't know. It's a little bit weak. Yes, but I will carry it. Hold it. So he was basically mixing flour with water. So when you do it underwater, you get dough, and if you continue to mix, then you will have the starch that will liquefy, and in the end, you get a very viscoelastic mass in your hands. And what he did, he left it over time, and he noticed that it was creating a very bad smell, similar to animal when it decomposes. So he called this matter plant-animal matter, in the sense that it was decomposing in a similar way of animals. Then these two chemists, they undergone very extensive chemical studies about this matter, and in particular, they determined the elemental analysis of this matter, and they noticed that it was made of carbon, hydrogen, oxygen, and also nitrogen, and sometimes sulfur and phosphor. They analyzed lots of raw materials, and they came to the conclusion that it was made with the same kind of molecules that was present both in meat, but also in bread, meaning in wheat, and in grass. So they had this idea that we have these building blocks that are coming from the plants to the animals, and that we needed to live. And this is where so it's Berzelius who advised Mulder in the letters, so you can find all the scientific letters between the two. And he proposed to call this matter proteins, from the Greek protos, because they thought that it was the very basic unit for life. And so this is Mulder, who said in the letter that the main form of animal nutrition that plants prepare for herbivores, which in turn supplied to carnivores. And again, here I give you some references, and it's quite fascinating to see how they went from this plant-animal matter to this idea that we have this structure in life that provides the nitrogen and sulfur sources. Then it has been started to understand what is the structure of this matter. And there, a chemist investigated the hydrolysis of glucoside with enzymes, and he came to the conclusion that an enzyme needs a very specific structure, like a key and a lock, to exert a chemical effect on a substrate. And so this was at the origin of the paradigm structure function of proteins. And there have been a range of investigation to find the structure of proteins. And this started with the X-ray diffraction on protein crystals, and the first structure that has been resolved was in 1958. From that time, there are many structures that has been acquired through either X-ray diffraction, but also with the cryo-EM recently, but also NMR. And the nice thing with this field, with the biological structure, is that they build a database from the 1970 where they store the 3D data. So there you can find all the 3D data that has been validated together with the metadata. So it's a very big source of information for structural biologists, but also for us food scientists, because we can dig into this database to find the structure, if it exists, of the proteins of our interest. So there have been a huge quest for protein crystals. And this is where physicists came into the game, because they were used to crystallization of inorganic molecules. And there they made this parallel between inorganic crystal growth and protein growth. And this is where they introduced the phase diagrams as a tool to help people to find the right conditions for crystallization. And they even, in 1988, went on under microgravity to promote the crystal growth in a nice way, compatible with X-ray diffraction. And then they tried to find a window with the second virial coefficient, so trying to find the right interaction potential to get the crystals. And this is the work from George and Wilson, in 1994. And so there have been quite a big theoretical framework to understand protein phase behavior for the crystallization, but not only. So for example, Ishimoto and Tanaka in 1977, they found that globular protein, lysozyme, they were undergoing phase transition like liquid-liquid phase separation through either nucleation growth or spinodal decomposition. So this is where they made the analogy with the molecular fluid. Like for example, this is the type of phase diagram that you have for argon, and also with colloids, and this opened a whole field of research to try to model proteins with colloidal particles. And they showed that by tuning the range of interaction, similar to what George says this morning, they managed to model the phase behavior of proteins. And so this has been a big field of research between the 2000 and 2010. And the main result is that they showed that the shape and the position of the phase diagram, you can relate them to the strength of interaction of your proteins and as well to the range of interactions. So protein has been seen, have been seen as sticky spheres. They built like universal phase diagram based on the second virial coefficient. So it's really a way to rationalize the phase behavior of any globular proteins. And sometimes, when you don't manage to get like the overall features, scientists needed to adapt the colloidal model, like adding, like for example, anisotropic interactions, and also changing the shape because it's not always very globular. So sometimes you need to add some specificities to model your behavior. So it was giving a very nice theoretical background for biologists to better understand the behavior of their proteins. But still, the biologists found out that there are a large portion of genes that code for long stretches, meaning like non-globular proteins. And so it was coming from missing electrons on X-ray diffraction pattern. And so this is how basically it looks. So here you have kind of globular structure that is fluctuating, but getting like a kind of globular structure. And you have these stretches that undergo an ensemble of conformation. And this may remind you the lecture from this morning with Peter with the polymers. And so you see that it's quite late, like 2008, so it's quite recent. And when you look back in the literature, it was already in, but with several keywords, like natively unfolded proteins, intrinsically unstructured proteins, intrinsically disordered proteins, and non-globular proteins. And right now, it's mostly intrinsically disordered proteins that is the main keywords. And so in the end, what they were analyzing the whole genome, human genome, and they noticed that only 30% of the proteins are fully globular, like the proteins that are very well modeled with colloidal particles. And you have about 10% of proteins that are completely unfolded, and you have the whole range in between. So you could think that here it's mostly like polymer-like or polyelectrolyte system. Here colloidal-like, but then you have everything in between. And this, it was also to highlight the limited knowledge that we have on proteins. So basically the proteins that are stored in the BTB database, so where you have all the structures, it represents only 4% of the whole proteomes. And they define like dark proteins in by analogy to dark matter, where you have some region where you can, by homology, guess the structure, but then you have also some proteins that we don't know anything about. And so for these type of proteins, like with intrinsically disordered proteins, this is where the polymer theory is very helpful. So here, this title is a bit catchy. So it's saying that a decade and a half of protein intrinsic disorder, biology still waits for physics. Actually, physicists, they were still interested in this type of proteins. For example, for gelatin, the polymer approach was already applied, but they were missing some general framework. And so there are Here I give the example of two teams, one experimental teams that is investigating, so in Zurich, it's Benjamin Schuller, and he's investigating with Fred the conformation of intrinsically disordered protein, and he's trying to find some signature of this disorder. So what they do, they determine the gyration radius, and they try to get this exponent. So again, a link with Peter lecture this morning. And what they show when you add denaturing agent, meaning that once you lose the 2D and 3D structure of the proteins, you get some, so for globular protein, well-folded proteins, with no denaturant, you get an exponent close to 1/3, so corresponding to a folded system. And once you increase the denaturation, then you go towards the coil conformation. And for fully disordered proteins, you see that you start with very high scaling exponents, and this could be related to highly charged protein system that can make a very extended conformation, and then you go towards kind of a plateau there. And then there is a team, but they are not the only team. It's just to show you the idea, but there are also people who are doing some molecular simulations, and for example, trying to see what is the effect of charge pattern on the conformation. And here you have the gyration radius as function of the charge pattern. So depending on if you have a very homogeneous distribution of charge, you can have very extended conformation due to repulsion. But then if you have like blocks of charge, it's expected to be much more like a folded system or collapsed system. So what are the recent advances in protein science? So you may have heard about AlphaFold. So it has been quite a revolution. So every year there is this CASP competition. So it's like the Olympic Games for protein structure predictions. So every year there are teams that are coming around and try to find the structure by prediction of a protein. And in 2020, this is where AlphaFold, 2018, sorry, this is where AlphaFold came into the game. So it's based on artificial intelligence and a company from Google. And actually, they really did a very good match as compared to structural biologists. And the second version was even better of AlphaFold. So right now, we are able to predict protein structure quite well. Not perfectly, but in a very unexpected effect. There are also some other ways. So this is a bit older, but there is this game which is folded, and where you can play around to find the right conformation of your proteins. So, and this has been a paper when the folded players was inside, meaning that the gamers were able to optimize the protein structure quite well, and they managed to uncover protein folding by simply playing around. So I can show you this. So for people who are working on protein, I think it's quite nice. So this, for example, it's a puzzle. You are being asked to. So this is the spike on the COVID virus, and you are being asked to find the right binder. So here you see the red. It's where you have clashes. So it's so much energy. You need to optimize the structure, and then you can play around to see how the conformation of this system should be, so that it interacts with the given proteins, but in a nice way. So it's not easy, but at least for you, it's a way to see that proteins, yes, it's not only, because when you draw the secondary structure of proteins, you feel like there are a lot of void, but actually no, it's full of atoms, and you have lots of steric repulsion between the residues. And I need to go back to my presentation though. And then experimentally, the major advance that has been undergoing in the last year is the cryo-EM. And this means that with cryo-EM, you don't need anymore to do crystals. You can directly image your proteins at a resolution that has never been so small, and that you can even get the details on the secondary structure. So this opens really a very good way for proteins that are difficult to crystallize, and it's the case for food proteins, for lots of food proteins. And this is the last predictor that has been launched by Meta. And so this was based on a different approach than Google. There they tried to read the amino acid sequence as it would be word. And then they connected this to structure. And so they managed to predict the structure. The accuracy is a bit less than AlphaFold, but then it's much faster. And so what they call this, it's like the protein universe with the unknown and known protein system. So it's a bit catchy, but yeah. And so now, yes. Okay. So these are the major advances in structural biology for food science. So it started at the same time as the discovery of proteins, because proteins were found in plants first with Beccari, and then there have been this scientist Osborne that has been a lot of work, and this book, The Vegetable Proteins, is very rich in information. And what he did, he tried to classify protein based on their solubility. And so he defined 4 groups of proteins: albumins, which are soluble in water and coagulable by heat. So it was an analogy with milk proteins, globulins that were insoluble in water but soluble in neutral salt solutions, prolamins because they were only soluble in strong alcohol, not in water, nor in salt solution, and glutenin, not dissolved in any on the other conditions. And it's still widely used in food science. Then food proteins research, it has been quite linked to nutrition, because there have been some recommendation for the daily protein intakes, and it was in relation to some disease that were affecting people, especially during the famine. And they came to the conclusion that the people need a certain amount of protein daily to be healthy. And this was what they call the protein theory. I won't go into the details, but you can find the references. And in 1950, following some very big famine in Africa, the FAO and WHO talked about a world protein gap. And there they identified that we really need more proteins to feed the world. And then there have been lots of research in technology to find a way to produce more proteins. And this is where actually the first textured vegetable proteins were produced. They also tried to make flour from fish, to produce proteins from single cell, and also to extract protein from leaves. And there is a kind of strange feeling now, because it was more than 50 years ago, and actually right now we are exactly in the same trend, so trying to find new source of proteins, and basically this is becoming again very trendy. This as well, this less from fish, but also texturized vegetable proteins. So if you work on this type of product, don't forget to look at the literature of that time. And yeah, this is just to highlight kind of a déjà vu system, and especially if you read this report from the European Parliament, there is they identify some new sources of alternative source of proteins, and actually a single cell production is one of the leads today. And then I will just finish with this. So it's a bit hard to see what has been done in food science and technology, because it's quite a broad field of research. What we have done recently is that we have worked with data scientists to do data mining in the literature. So we collected publication from 1980 to 2018. And we try to figure out how on pulses, because pulses are a good alternative, for example, for meat, and but it's still not used as food. And how is structured the field, and this I will just focus on proteins. So if you look for, so there are papers on biochemistry and structure of proteins. Then also on their functional properties, like interfacial properties, gelling properties, and also on their digestibility and allergenicity. And if you look at the time frame, basically, if you want some information on the biological properties, molecular properties of proteins, you need to go to this frame time. And then lately it's more about functional properties and also as function to replace gluten. So for this first part, the take-home message is that the protein research, it's a mix of many disciplines, but it's true for many areas. So there are lots of structural biology that are advancing a lot on protein structure. First, it was a chemist who tried to find out the chemical structure of proteins, and the physicist who provided some theoretical framework to better understand the protein phase behavior. And then for people who are working with food proteins, it's very intricate with nutrition knowledge and technologies. So in practice, if you want to find some relevant information, I would suggest, on food proteins at least, I would suggest that you look into the large source of information from the beginning of the 20th century, because there are lots of things about solubility and behavior. In the 1980s, there has been large amount of work on biochemical and molecular properties, and in the end, 2000s, it was more about techno-functional properties. But don't forget the old papers, because there you can find very interesting information. So now I'm getting to the amino acid composition. So food proteins, I will be now focusing on food proteins. So there are very various intrinsic structures. So you can have globular proteins, like for example lysozyme. You can have non-globular proteins, like wheat glutenins, for example. And you can have some model in between that are both globular, but also have some disordered domains. So not fully disordered, not fully ordered, but but in the middle. In the raw materials, they can be either as a monomeric state, and mostly, for example, if you take egg white, it can be quite monomeric. They can also be in the form of clusters, like for example, casein micelles in milk. And when you take seeds, they are in very dense system. So you have some native assemblies that can vary a lot depending on your raw materials, but also depending on the history of your system. And then you add processing. And there you change the temperature, the pressure, you apply shear, you change concentration, etc. So basically you tune the protein structure, but also the interactions, protein-protein interactions, but also protein and other compound interactions. And then you get some functional networks. So you can have colloidal gels, fibril gels, but also the 2D network at the interface of air and oil, but also air and water. I don't know what I said. So it's quite diverse. And so what we wanted to do, and this is a bit focused on what we've been doing, is so in biology you have a link between sequence, structure. Yes. It depends. It depends on the process, and it depends on the proteins. With globular proteins, you can create gels with simply phase separation, and there you keep, it's basically you're creating a big continuous network with a glass in the dense phase. But sometimes you lose completely the structure, especially if you put temperature above the denaturation. Then the theory that was applicable for the native state is not applicable anymore. So every time you add something, it's getting a little bit more complex. So the idea here was, could we find a sequence functionality relationship? Because right now, when you have a proteins, you need to screen. So basically, you need to go through all the steps. So you purify and you probe the functional properties. Our idea was to look into the sequence and see if we can find some critical determinant of the functionality. So what we did, so we consider the protein sequence. So here you have a way to code the amino acid sequence. Each letter corresponds to one amino acid. The structure of an amino acid, there is a generic structure. So here you have an amine function, a carboxylic function, and what makes the specificity of an amino acid is the structure of this residue. And so, for example, if instead of this letter I plot the fluctuation of the molar mass along, you see that you have a fluctuation with a minimum molecular weight with glycine, which is the simplest amino acid, and tryptophan, which is the biggest. And so you can guess that being a side glycine versus tryptophan is not exactly the same. And you can, so this is just for molar mass, but you can also do it for charge. And then, for charge, and this is, for example, for these proteins, how it looks. So here you have negative charges, and here positive charges. And you can do it for any properties. So this is what we did. We tried to convert this sequence into all the physical chemical properties that we could find. And for that, we needed the sequence. So we went into the open access database to get the sequence of food proteins. And then we got the theoretical paper to calculate a number of parameters, like specific volumes, refractive index, water sorption, because in food science, it's quite important to see how a powder would behave depending on the humidity. So what we did data mining in the in Uniport dataset. I won't go into the details, but we can discuss it later. Then we cleaned the protein sequence and we annotate them. And then what we did, we developed a tool which is called Protein Pred. So this may be of interest for people who are working with proteins. And actually, who is working with proteins, just to know? Okay, some of you. So this tool, you can use it. So basically, you can have 3 types of inputs. Either you have a single amino acid sequence, multiple sequence, or if you have the amino acid composition, the experimental one. So for example, if you have a single amino acid sequence, this is how it looks. So you can load an example. So this is a proteins napin. It's a small proteins with 2 chains and 4 disulfide bonds. And then you compute. And what you get first, the first is to get the number of amino acid per proteins. You get the chemical score. So for proteins to be nutritious, it needs to have an equilibrated amino acid composition. And so this, it tells you whether, if it's above 100, it tells you if your proteins is kind of complete or not. And so here you see that we have a limiting amino acid leucine, meaning that in your food you need to have proteins that would bring leucine to be equilibrated. And then the type of parameter that we compute, it's so we have the molecular weight, the isoelectric points. We also have extension coefficient, partial specific volume, and all these type of things that are helpful when you probe experimentally proteins. And for example, UVV spectra, and also water absorption for the proteins. So we did this for all the proteins that we gathered, and we built this database that is also freely accessible. And with this, for example, if you look for napin, this is the same proteins that I showed you later. So you see that actually it's not only one proteins, it's a bunch of proteins. And when you have a look on the properties, like for example the charge, you see that it's it's the same name, but it's quite different. So depending on the sequence, you really have different properties. So meaning that when you have a protein, it would be nice that you identify which one you have, because based on general properties that you have on one protein, this may not be applicable for your protein. And so then you can look. So this is a way to look at the database, is how the proteins are composed in terms of amino acids, and how is it in comparison to FAO recommendation in terms of essential amino acids. So the recommendation of FAO, it's in red for essential amino acids. So you see that most of the proteins are okay for this, but for example, for these ones, Yeah, for this one, for lysine, for example, it's quite broad. So you have quite a lot of proteins that are not completely equilibrated. So, and this is the case, for example, for wheat. And this is why you need to complement wheat with pulses, because they are complementary in terms of amino acids. And so what we can see, so here we map the food proteins that we have, the major ones, in terms of degree of rigidity. So here you have quite rigid, the prediction of quite rigid proteins, here quite flexible, and here as a function of an hydrophobicity scale. And the size of the dots, they are proportional to the charge of the proteins. So the smaller the points are, the less charged they are. And what you can see here is here you have most of the globular proteins from milk and egg, meaning that they are expected to be quite folded. And there, it's like, for example, wheat proteins with prolamins. So they are expected to be not very charged and quite flexible. And you have a whole bunch of proteins that are in the middle. And the nice thing is, like here in yellow, it's rubisco. So it's the protein from leaf. And experimentally, we've shown that it has quite nice gelling properties, and it was behaving quite similarly to egg proteins. So it seems like the amino acid sequence could give an insight into the behavior of the proteins. And it's quite easy to have access to the amino acid composition. So here it's only what I showed you, it's only like average properties. But actually you see that there is some interest to also check how the properties is distributed. And from that point, so we are not there yet, but the idea that we want to test, and this is from the structural biology community is to consider this as a signal, to decompose it using, for example, a Fourier transform to get the distribution of the properties and to see if there are some motifs that can code for the functionality. But this is more like an opening. So here I talked mostly about the primary structure, but there are also data on 3D structure. So for example, with 3D structure, if you have all the coordinates of your atoms, and this is what is provided in the PDB, you can compute the individual pKa and then probe the electrostatic on the surface. And so this is an example for Rubisco. And so if you don't take into account the structure of the proteins, is this one. And if you take the structure into account, the 3D structure, then it's the dots. It's the triangle. So there is a small shift depending on the structure of the proteins of the properties. And if you look into the electrostatic map, you see that here it's quite homogeneous, with negatively charged on one face. Yeah, it's maybe not the best example, but I will show you later on this one. So here is just an example of two proteins. They have the same size, the same isoelectric point, and same charge, experimental charge over the pH. But if you look into the details, you see that the napin, it have a very big patch of negative charge here, whereas for lysozyme, it is mostly positive. So here it's global charge, but actually there is a big patch of negative charges. And what has been done theoretically, especially by Michael Lund, it's that the patches, it provides some orientational interactions, and it can provide the assembly of proteins and favor actually attractions. And it was used to explain some results from a lab from Rennes, who showed that two isoforms of beta-lactoglobulin, B and A, and it's only one amino acid that is different between the two proteins, the interaction with lactoferrin was completely different. And so they interpret this data based on the fact that the mutation for one amino acid, it was inducing a dipole and was very much favorable for the assembly. So from our result, what we were expecting with lysozyme and napin, since napin has a very big patch, we were expecting to have a strong assembly with an oppositely charged protein. But this is not at all what we observed. So with lysozyme, we got a big response. So we have assembly with an oppositely charged proteins, but with napin, nothing. And we came to the conclusion that maybe it was related to the flexibility of napin, where we have all of this disorder inside the globular structure, and but we still need to go deeper into this. So if you don't have your protein structure, you can use AlphaFold or ASMFold, so predictive tools. What can we learn from that? So for example, for lysozyme, which is a quite globular proteins, you have quite a perfect match between the prediction and the experimental data. But it's probably been used as a training set, so it's maybe like kind of a normal. And then when you have a partially disordered proteins like gamma gliadins, we see that with the two, we don't have any structure for this because it's not possible to crystallize it. And so you see that on the part where you have some 2D structure on the C-terminal domains, the predictions are quite similar, but then you have all these disordered domains that are actually quite that are not well predicted because it's probably fluctuating. So just something that you have to be aware if you use these tools is that these tools, they don't take into account the post-translational modification, and this can be quite important for the structure, especially for food proteins. So it doesn't take into account the hydrolysis or phosphorylation. And also, if you use the basic version, it doesn't take into account the oligomerization state of your proteins. And for special proteins like this one, experiments still work. So then you need to purify the proteins and characterize them experimentally. And this is the type of study that you can do. So with the purification facility that we have in Nantes, we prepared some purified gliadins, so the proteins with the two domains. We purified the full-length gliadins, the N-terminal domain that is expected to be disordered, and the C-terminal domain, which is expected to be folded. And if you characterize them with biophysical tools, for example, with circular dichroism, we see that the N-terminal domain, it has this shape that is compatible with intrinsic disordered proteins, while the C-terminal domain, it has this signature that is quite attributed to secondary structure, especially to alpha helix. And then you can use also X-ray diffusion, small angle X-ray diffusion, scattering, and then and you have signature either for folded domain or disordered domain. So basically, when you don't have the prediction, you can go to experiments and try to find out what is the signature of your system. And I won't go into this because I think I will be running out of time. So for this part, get to know your proteins before you test them, because you can have already quite a lot of information based on your amino acid composition. It's most of the time it's not enough, but it's already a good way to start. And literature, of course, but this was the first part of my talk. Okay, so now I'm coming to the physical framework to model proteins. And so this will remind you the 2 lectures of this morning. So for proteins, which are quite in a globular manner, you can model them as colloidal particles. So here I put a sketch of the LVO theory. So you have electrostatic repulsion. I don't have the secondary here, minimum. And here the attraction at short range. What people have been doing in the protein science field is that they were thinking in terms of second varial coefficients, meaning that the integration, the area under curve, and for B2, so second varial coefficient positive, you have repulsion, and when it's negative, then you have attraction and assembly. And for proteins that lack any structure, then you can use the polymer approach. So either like for linear neutral proteins, but also neutral polymer, but also you can increase the complexity by using polyelectrolyte theory. And so this is for sure the lecture from this morning were very much into this. And this will remind you as well, but maybe in more simplified way, the phase diagram in 1D as function of volume fraction. So when you have proteins that is well modeled by a hard sphere, and then if you add attraction, then this is where you can have gels. And if you have repulsion, you can also have some clusters and some, for example, repulsive glass. And for when you want to use like polymer or polyelectrolyte science as a framework, so this is again what has been, so it's very nice to have my talk today because it's quite fresh. So here it's for neutral polymers, but then if you have proteins may also be seen as polyelectrolyte because it has quite a lot, can have quite a lot of charge along the sequence. And there is more the theory about polyelectrolytes that apply. And so in the field of proteins, now our physicists are really into it, and it has been quite an intense field of research lately, especially that because these proteins, intrinsically disordered, they have been shown to undergo phase separation, and especially in vivo. So this was quite a breakthrough in biology, meaning that you don't need membrane to compartment. It seems like only phase separation, pure phase separation, it's enough to get a compartment, a dynamic compartment in cells. So it's quite active field of research, and so they are trying to get what they called the grammar of phase separation, meaning to know if we know the amino acid sequence composition and also distribution, can we predict the phase behavior and then the stability of the system? And I think that for food science, it's a good inspiration because it would make our approach much more generic rather than specific to one protein system. So if we go back to this, for food science, in first approach, we could think that here we have the 2 extreme with the colloidal model here, and it's actually working quite well for protein from milk and egg. And here, it's working quite well with neutral polymers. But then in between, as I already said, you have all the intermediate that are possible. So for example, you can add stickers on the particles. You can have spacer and stickers, and depending, and this will also remind you this morning, like the pill network. So I will show you the example of 2 ideal candidates. So if you are run into this, you are lucky. So the experiment is like we do osmotic stress, meaning that we put the proteins in a dialysis bag, and outside we put the stressing polymer. then the stressing polymer with the osmotic pressure that is higher than the protein dispersion. Then over time, you will have a migration of the buffer and ions through the semi-permeable membrane, and you will get a concentrated protein system. And in the end, at equilibrium, then you have the protein concentration inside, the polymer concentration inside. So what you have is the osmotic pressure as function of protein concentration. And this is the two ideal candidates. So here you see this is the alpha-lactalbumin, so proteins from milk. And here are the dots, and they probe the osmotic compression for several ionic strengths. And actually, they were able to model it quite well with a hard sphere model and Kernel-Sterlin equation, meaning that this protein, actually on the range of concentration that is probed, is quite well modeled by a hard sphere. And here you have sodium caseinate, so it's a mixture of caseins, but not in the form of micelles. And what they saw, so in relation to the previous talk, they saw power laws, if I'm not wrong, and two regimes. And then you can explain this with physics and then transfer it into scaling law. But basically, so they found that these two regimes, with here you have the overlap concentration. At large concentration, you have a deviation that has been attributed to the contribution of contour ions that are in the dispersion. And the deviation is also in the deviation that you get some complexity, but sometimes it makes the specificity of your system. So if you're a PhD student and you come into this, you are quite happy, but usually with food proteins, you never get into this. Usually you have deviation. So this is, for example, work from Rennes, from a lab in Rennes, where they studied the osmotic compression of lysozyme. And what they observed first, as compared to a hard sphere in a continuous line, in the first range you have repulsion. And then you have this plateau, and this plateau was related to crystallization. So basically, they probed some. So there was some repulsion in the system in the first regime, and then you have a plateau, meaning that you have a phase transition between the 2 points. And here you have crystals, and then it's getting very hard to compress the system. And this is for the casein micelles, and it's not following at all the prediction for colloids. And it's actually here it was related to small peptides that contributed a lot to the osmotic pressure, and here to the resistance to compression due to hydration. So you can do osmotic stress experiments, and then you compare it with theoretical model and try to explain the difference that you have from the models. And in the literature, there are also data on BSA, ovalbumin, and gliadins. Something that we always wonder whether protein is colloidal or polymers. And on one system, there was this nice behavior. So this was during purification. Actually, it was during my PhD, so quite a while ago. So I'd used the housebound procedure. So I tried to extract albumin globulins in the first 3 washes, and then I got the gliadins that I was interested in detatol. It was late Friday. I left everything in the fridge, and on the Monday later, I came back and I had this layer on the bottom of the tube. So it really resembled to honey. And then I discovered that my literature review was not good enough, but because actually it was observed in 1925, so I'm going back. And they observe indeed, like if they hold the solution at 5 degrees, actually it phase separated. And this was the first proof of liquid-liquid phase separation of wheat proteins upon decreasing temperature. So from that, and the nice thing, because the first thing that we do as a biochemist is that we check the composition in proteins. So we do this with size exclusion chromatography. So here you have the size distribution of the flour, of the extraction. So you see that we have big polymers, like it's above 100 kilogram per mole. And you have the small proteins that I was interested in here. And upon the phase separation, actually, I removed all the big ones. So for me, it was a very easy step to purify. And at the same time, it was funny that it was only the big ones that were affected by the temperature. So what we did several quench. Then after this, it opened a whole set of studies. And what we plot, we use Florey-Huggins theory to plot the distribution of the proteins in the upper phase and lower phase as function. And we plot this as function of molecular weight. And what we've seen is that here, for the small proteins, the molecular weight was not important in driving the phase separation in the range of temperature probed. while above a critical molecular weight, then you have really a behavior that resembles to neutral polymers. So it seems like we thought that we identified a boundary that would separate colloidal-like behavior and polymer-like behavior. And then a team in collaboration with Laurence Ramos and Amélie Ban, who are there today in this summer school, they went further and they analyzed with the more complex system and tried to see whether you can find a generic behavior. And for glutenins that are quite complex, actually they are quite well modeled by Florio-Gil's theory. So it's a good way to simplify a system that we think that is very complex. And then I refer to the lecture of Amélie next week, who will talk about about gluten. So the take-home message about this part is that protein may be complex, but the use of soft matter physics, it can help in describing behavior. It offers a set of tools, of course, to map their assembly. Here there are two, but actually you will see for the whole summer school, I think rheology and other things are very interesting to better understand our system. And just this was in 2015, so there is this big research about applying colloidal theory to proteins. And this paper in 2015, they criticized the colloidal theory for modeling globular proteins, showing that proteins, they undergo conformational change. So then the colloidal theory may not be applicable in all cases. And yeah, it has been kind of a battle. And I guess editors like that. Because then there have been a reply from this group and try to see that maybe the colloidal theory may not be enough to describe all the specificities, but sometimes it's enough to get the behavior that you are probing. And this is an illustration on how to model proteins. So here you have the proteins, the real proteins, rabbits, and how you model them. So sometimes you need this, but sometimes it's enough just to see the blank as like non-interacting amino acid, positive, negative. Sometimes it's you say that okay, it's a sphere with several spots, maybe a little bit anisotropic, but sometimes also it can work that it's only a hard sphere. It works. So maybe start from so I'm really not I'm not as experienced as these ones, but I would suggest to search for genericity, even though you probably will get specificity, but it's maybe in the specificity that you will find some interesting properties of your proteins. And finally, the last part. It's how to go from model to real protein ingredients. So there, the step is quite big. So first, I feel like the tools that are provided by soft matter physics are interesting to probe protein structure, especially in concentrated systems. So here so I focus on plant proteins because it's kind of my field, but you can really apply this to any food sources. So for example, you have, so proteins in plants, can be in grains, but also in milk it's in a colloidal state. They may be in native state, in native in native assemblies, meaning that it's not the native proteins, they are already assembled. And they may also form like stabilized oil bodies. When you apply processes to produce the ingredients, you have some steps like precipitation, ultrafiltration, defiltration, drying, like making the system that already before putting some temperature shear, it may already change the native assemblies, but also the intrinsic structure. And then if you if you use this ingredient to create food, then you have sometimes, if you start from dry ingredients, hydration, interfacial stabilization, and network formation. And this is the images. And all this grow through the processes. And so there are many questions about the structural transition during the food processes. Is it reversible or not? What is the structure in the dense phase? Because most of the functionality of proteins, they are occurring in dense phase. And where is the water, and what is the effect of water on the structure that we observe? And so for this, for sure, osmotic compression, and this a very nice job from Antoine Bouchou. They used osmotic stress experiments to understand the behavior of casein micelles over a large range of concentration, and they showed that they are actually like deformable, compressible colloids. But for this, you need to combine osmotic stress. So this is our experiments on rapeseed cruciferine. And you need always to combine, like, for example, osmotic pressure and Sachs. And here we see that with increasing concentration, we start to see the structure factor of the proteins, but then in very high concentration, like for us it's quite high concentration, 500 grams per liter, then you really have a compression of your system with the shift of the maxima. So this is just to still open question. So there are some osmotic data in the literature, and the funny thing that we see is that when you overlap all the data from different, it seems like in the last regime we get quite a similar power law, let's say. But right now what we miss is that for this we need a lot of proteins. So we need to purify grams of proteins to do this type of experiments. And I discussed with some people who are doing numeric simulation, and he told me, come back to me with 150 points, and then I can model your data. But for us, it's very tedious. But there are things that are moving now because people are developing some microfluidic chips to make osmotic compression. And this open routes to get this osmotic curve for a various range of salts, of temperature. And maybe this will be a way to see the genericity and if this could be related to the hydration of, for example, of the hydration of the system. And this is to relate some bulk properties with interfacial properties. So it's two sets of experiments. This was done during my PhD with the osmotic pressure as function of volume fraction of gliadins. And this is experimental data from Aurélie and Laurence Ramos group, where they probed the interfacial properties of these proteins at water-air interface. And what they notice is that in terms of surface pressure, they observe some transition that I observed in bulk. So for proteins that are forming monolayer, there may be a link between bulk properties and interfacial properties, and it's maybe a way to better understand the 2D behavior, or the other way around. Then what can bring the model systems? So one of the problems in food science, and this may be something curious for you, is probing protein solubility. So there is no standardized way to probe protein solubility, and actually it's maybe more the colloidal stability that we should talk about, but in food science we talk about solubility. And basically what we do a partial solubility. So we hydrate water, we hydrate the powder, we wait until the long time enough so that it's in equilibrium. We centrifuge. Every lab has its own protocol, so bigger the speed and duration, and then the relative, what remains in the supernatant is considered as the soluble part. You divide by total, and this gives you the protein solubility. So depending on the conditions, you never get the same type of data. So maybe a way, and this is inspired by the field of pharmaceutics, where they also have some problem with colloidal stability. For example, when they want to make vaccines and high concentration of antibodies, they have some phase transition. So it's also a question of how to probe solubility of high concentration of proteins. So in theory, solubility, it would be the concentration of protein in a saturated solution that is in equilibrium with a solid phase under a set of given set conditions. But it's hard to avoid an equilibrium state. So there are two approaches that are proposed but not used yet in food science. First is to use depletants. So as George introduced it this morning, the idea is that you put a polymer in the dispersion and then you check you induce phase separation and you probe the protein concentration in the soluble part, and then you extrapolate for 0 depletants, and this can give you a way to check the solubility. The other way is to probe the phase diagram, and so then you can really have a map of the solubility as well as the state of the system. And so you can have some nucleation growth and spinodal decomposition, and yeah. So there are systems that has been developed to probe this type of phase diagram. So for example, with microplate, with a control temperature chamber. There are also millifluidics. So then with this, you can really increase the throughput of your analysis. So basically use connectors that you use in chromatography. You make a drop, which is your sample, and then you tune the properties. And this is a way you probe the gray level of your image, and you can plot the phase where you have monophasic condition and biphasic conditions. So it's easy, and it's quite easy to scale up. So to probe in a maybe not high throughput, but mid throughput phase diagrams. And the last part, because the phase diagram, it's also about kinetics, and what we developed recently, it was based on microfluidics. What we built, we built giant unilamellar vesicles. This, and inside so with phospholipids, and inside we put a protein which is called hemolysin. Hemolysin, it's toxic proteins that is able to bind and to form some pores on the cell membrane, and it's actually a toxin. It's a way for the bacteria to kill a cell, and so this we introduce them in our bilayer, and in that way we can exchange. We can keep the protein inside, but exchange the small ions. So it's kind of a dialysis bag, but at a micron scale. And with this, we control very well the flows, and in that way we managed to build this phase diagram, including some dynamic data, especially with spinodal decomposition, because we can go very fast from pH 4 to pH 6.5. So then we can go to the dynamics of phase separation, and it can be interesting to get some structures, because this has some functionality in terms of viscoelasticity, for example. Fini? Okay. So it's the end. So just for you to see the okay. So when you fraction, so this is also to show you, so the fractionation, the way you create your ingredients. So for example, from seeds, you can make concentrate in the dry state, but you can also go through a wet route, meaning that you solubilize and then you dry. What has been shown in the literature, and this paper nicely reviewed this, is that when you go through this route, it induces a lot of aggregation. And especially is the precipitation and thermal treatment that induces a lot of aggregations. So you can go to dry fractionation, and then you get a concentrate that is much more, let's say, functional. And this is a result from this PhD student that is finishing her thesis right now in our lab. And she saw that, yes, there are proteins in these ingredients, but there are also many other things. So usually we have a focus with proteins, and we want to apply lots of theory, but actually in the ingredients that are in the market, we have, for lipids, so it's in yellow, we can have up to 10% or 12% of lipids in addition to the proteins. So, and she noticed that with the weight fractionation, there have been an accumulation of lipids in this type. And what she thought it showed, is that the lipids in ingredients affect a lot the mechanical properties at oil and water interface. Just for you to know, she is looking for a postdoc, so if you have any position available, she's a very good student and and she brought a lot of things in the field. And more details will be given on this type of topic by Yolaine, who is here, and will present a poster on Friday, especially on the effect of fractionation on the composition of these ingredients. And so take home message for this part. For sure, there is a set of tools that we can use in food science, but still we need to characterize our samples as thoroughly as possible, because sometimes there are some minor compounds that can have drastic effects on the properties that we probe. And this is it.